So as Manju ma'am has given the content or the topic for today's day is protein-protein interaction. So before going exactly into the topic, let us try to understand what is a network. So can anyone tell me what can be a network? In simple terms, you take for example yourself. What sort of network you have with the others? What sort of who will be a first, if I say network, who will be the first close person to you will be your parents, right? So you are having a very close relationship with them. So you're creating the first network, that is your parents, whom you meet daily, minute to minute, second to second, talk to them, right? Then what comes the next set of network you're going to have? Maybe your siblings comes next if you're having them. Okay, so they are also close to you, right? Okay, now let us extend the network little bit. Hope I am audible. So coming to the next network, what is the next network you have? Maybe your neighbors, if some of them are there, they may be some close neighbors to you. So now when I compare the network, what you have created with your parents, with your siblings, little bit different when I take your neighbors, right? Okay, now go to the next network, what we are having now. Parents, siblings, neighbors. Now go, let us assume you are going to a college. You're all, many of your college, right? So when you go to the college, you'll be having some friends, maybe 10, 15, 20, 30, maybe. Okay. But when I take the numbers, is a network, is an interaction between you and those people. Is it similar to that what you have with your parents or with your siblings, or it is with your neighbors? It is little bit different. Am I right or wrong? If I'm right, just put Y, capital Y in the chat box. Okay, I've got one response. Yes, yes. Okay, so yeah, fine. So so now you understand what has happened. There is, yeah, exactly. Yes. So everybody are accepting with me? Fine. So what has happened now? You had one particular source, that is yourself, and now you are having a network. Okay. The network between you and with the others is not of the same distance, right? Some is very close to you, some are little bit away from you, some of them are very much away from you, right? So similarly, taking this as an aspect or an idea, I am going to talk to you about the today's topic, which is called as the protein interaction. So it's what I gave you as an example is a human interaction, but we are trying to correlate it with the protein interactions. Okay. Do you think that proteins and proteins talk to each other inside a biological system? Why? I expect why in the chat box. If my participants are active, so I should get like this the answers, right? Yes. Okay. 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 Yes. So, yes. So there is an interaction, there is an talk happening between the proteins which are present inside the biological system. Okay. Now, yes, proteins are talking, interacting. Fine. Thank you. So we have to see how much is that interaction. Is it a very strong interaction or it is okay, less interaction, or it is a very, very weak interaction, or some proteins may not interact also, right? So keeping all this idea, some person in USA, his name is called as Stanley Milgram. He is a social psychologist. So he was doing an research and he did an experiment of trying to understand what is the minimum number of network or interaction, minimum a person can have, a human can have with the others according to his experimental results. He did a small experiment on that one. Minimum to minimum, he has got a result which says that on an average, an individual among the human population, they will be expected to have 6 number of networks or interactions within them, okay, or among them. So with this idea only, our mathematicians have created one algorithms. So with that mathematical algorithm, they are able to create a network to understand about a protein, how it is able to interact with another protein. So is it very close interaction or is it a very less interaction? and so on. Okay. So, okay, the topic is protein-protein interaction, the basic I told you. Now, when I have two proteins with me, by just drawing on a sheet of paper, I know that, yes, this is one protein, this is another protein. If I keep them apart, I will tell there is no interaction. But if I try to draw one single line between them, what we will understand, yes, there is some sort of understanding between both the proteins. They may work together for a positive function, or they may work together for a lethal function also, that we don't know. Okay. So two proteins you're able to draw on your paper and show it. Let me assume I'm giving you 10 set of proteins. I'm telling that this protein is linked, having interaction with this is having with that is having with this, like this. I'm giving you multiple set of data. You will tell, ma'am, only 10 proteins. No worry, ma'am, we will do it. We are all excellent in doing that one. Tuck, you will take one paper, draw it off, get me the result and tell, ma'am, these are the number of interactions for each and every protein. Okay, fine. Two proteins worked out well, 10 proteins worked out well. Now I'm giving you 1,000 set of proteins. Now what we will do? Can we take a piece of paper, draw everything and tell which protein is interacting with which one? How much is the strength of that interaction? Can we do it? Maybe not possible. And that is the place where our tools, computational tools are coming into the help to tell how much our proteins you take, how much amount of interactions they're going to have with the other proteins. Okay. So there are different databases by which you can check the interactions and find the networks present between the protein to protein. Today too, I will show you. One is a string database. It is a free online software tool which you can use and understand the network. If you want to little bit explore more about the networks and visualize all the details very clearly among the proteins which are interacting, we go with a little bit more advanced tool of it, which is the Cytoscape. Now, I'm not going to separately talk to you about string database and Cytoscape, but I'm going to link you between string database along with the Cytoscape tool to visualize the protein-protein interactions. Okay, so this with small note, I'm going to share my screen and try to explain to you the theory behind what is happening during this. Okay, so I think my screen is visible to you.
Yes, ma'am.
Yes, ma'am. Yeah, fine. Thank you. So this is the topic for today, Cytoscape, a tool to visualize protein-protein interaction. So what is protein-protein interactions? In short form, you call it as PPI, which are physical contact between two protein molecules. Okay. Now, these interactions are essential for many biological processes that include cell organization, signal transduction, as well as for immunological responses. So when I've given all these terminologies, maybe background in your mind, there will be, okay, ma'am is talking about cell organization. There are some proteins inside the cell which will pass the message and then the information goes inside. Similarly, if you take immunological responses, antigens are there, antibodies are there, you have different other proteins present in the system which also will be coming under this, right? So they all are interacting, talking with each other, passing a message and trying to perform either a positive reaction or a negative reaction depending on what type of the protein has been interacted. Okay. Now coming to the importance of protein-protein interaction. One of the main reason for people to study about this protein-protein interaction is to study about a disease pattern. Okay, so it can be remodeled during a disease state so that by understanding them, you can know the importance of that particular protein which is involved either to study about the disease progression, or you can design some protocol or a treatment pattern for that particular disease by trying to predict what are the proteins and the other protein which is involved in the interaction during the disease state. Second place where we can find the significance or the applications of this protein-protein interaction is in the drug design process. Okay, so PPI can also be used to identify potential drug targets such as nodes, okay, that are highly connected or which will be affecting a single disease pathway. So depending on what type of a drug target you want to, you can take some set of proteins and work out them, which was the hub, that is a main potential gene or a protein, and then from there you can start to design certain drugs for that one. Okay, we'll come to that step by step. Then understanding the gene function. So protein-protein interactions can help researchers understand the function of a particular gene product if something it is new to us. Okay. How can you study the protein-protein interactions? There are some experiments which are being done in order to, that is your wet lab protocols by which you can understand the protein-protein interaction. And extension of that one will be your computational analysis. The first method is called as co-immunoprecipitation method. It's one of a very popular technique where we are going to take two proteins and allow them to form a complex. And after that, you're going to synthesize a one single antibody, which will be specific for that one. Out of two proteins, it will be specific only for one protein. Okay, so that it will bind to that one, and based on the strength of protein-protein interaction and that recognized by the specific antibody, signals will be coming out, and you are able to get the best protein-protein interacted complex. Next one is called as Y2H, which stands for Yeast 2 hybrid system. So this is also used for analyzing or investigating the proteomes of pathogens like hepatitis C virus. So here what we do is that we take a transcriptional factor, allow it to bind with the specific proteins for with which it will interact with. So binding of that one will pass some message, or they will change some patterns of the transcriptional factor, which will be linked with an a protein which gene which is called as the reporter gene. So the expression pattern of that reporter gene, the strength of how the reporter gene is expressed, you can estimate the interaction between the two proteins with the transcriptional factors. Okay, then computation. So these two are basic protocols, are the highly advanced, and second one is basically for the molecular biology experiments they use it. So very advanced techniques they are to do the wet lab experiments to identify the protein-protein interactions. So based on these things, you are getting now the computational analysis. So we have different sort of PPI networks with which can be created, generated, and you can identify the novel proteins present among your network. Okay. So for this computational analysis, we are dependent on the data sets that are available in the different databases. So depending on what is your work area, your interest, your target, from based on that one, you can go to your database, get the source, and then find the better targets for your interaction process. Okay. Now, when we want to understand about the protein-protein interactions, simple terminologies are there, which let us see what they are. Now, this is a small network, what I have kept in front of you. There are certain terms over here. First term is called as the node. So now you can see that node is given in circle as its design or a structure over here, and it is going to be green in color. So the you can change the colors also. So here for this particular diagram, here in this network, you are able to see this one. This is the node. So can you tell me if this is defined as a node green in color, can anyone tell me or type in your chat box for this particular picture, how many nodes are there? It's a simple network. So how many nodes are there? Answer. Chat box. Are you using chat box? Am I audible?
Yes, ma'am, you are audible.
Then why people are not talking to me? Okay. So you have typed over here. So I have to keep it So the numbers what you have written is coming to 11 and 7. Mehbooba, just to check out the picture, is it 11 or is it 7? How many are there? I've shared the screen again. Slide is there in front of you. See, check out properly, ma. How many are there? You check out. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So all together in this particular network, we are having 11 nodes. 11 nodes is equal to 11 proteins coming from 11 different genes. Okay, so you can see that so many are there. Now in the center, okay, in the center, it was a node, but that node we have converted instead of a circle to a square, and we have changed the color also. Okay, and what is that one called as? What is the name given to that one? It is given as hub. Okay, so whenever you are creating a network, you can extend that network to understand among the different nodes. What is a node? I told you it is the protein. Okay, coming from a gene. So that particular protein is having how many interactions with the other proteins. So you said there are 11 nodes over here. Even the another one, this square is also a node, but since it was not circle, you could not identify it, but that is also the node only. The thing is that when we want to highlight which is the potential node, which is having lot of interactions with the other nodes, for explanation sake, we have changed from the circle to a square. So actually in this network, we are having 12 nodes, including the square also. It was a circle earlier, but to highlight its importance, we have converted from circle to a square. Okay. Now, why did we do that one? And I've given here that it is called as what? It is called as a hub. Okay, so this is called as a hub. Okay, so they're called as a hub genes, which are the potential genes which are having good, strong interaction with the other proteins. Okay, or other genes. Fine. Okay, now you can see that the second term, what you have in this picture is called as an edge. Okay, now edge, is it a circle or is it a straight line? It is a straight line. Now, when you are seeing that one, you can find that the edge is connecting one node with another node. So you can check out the edge is connecting one node with the other node. So if that is the case, can you all tell me how many edges are there in this particular network? How many edges are there in this network? Can I get the answer? Is it coming? 13. Purna, check once. You have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. My count says 14. Did anyone type 14? Yeah, so that is 14 edges are there. Okay, fine. Now you concentrate only on the hub genes or the center one and tell me how many edges are there for this one to its adjacent nodes. Just count and tell me. From this hub genes, how many networks are there or how many edges are there? 6. Okay. So it is 1, 2, 3, 4, 5, and 6. Okay. Now you said 6, right? Then why did, so that's why we put this as a hub genes, right? Now you take this one also. Can this be a hub gene over here? It may be having it, but the interaction is less because it's interacting only with 1, 2, 3, and 4. Here it is interacting with 1, 2, and 3. Here, if you take it's 1, 2, and 3. Here you take this is having only one interaction. So among all the nodes, what you have got, this is one of the node which is having the maximum interaction with many other proteins of your study. Okay, so that is a reason why this particular gene you call it as the hub genes. Okay, how to get that one we will see later. Now this is for you. You can see this one. Can you all tell me how many number of nodes are there? I removed my PPT. Yes, visible. You have typed the answer. 6, huh? How many are there? How many are there? Okay, 12. You're answering me through the chat box. Right, fine. Okay, now coming to the, so how many edges are there? It's a very simple network, right? So you have written 13, fine. Okay. So similarly for the other one also you can see. So what I'm trying to show you is that this is the small network, right? Minimum number of genes, the network is very small. But little bit when you increase the number of your proteins, what was happening? The network is increasing. Okay, so that is the reason why instead of going with a technical way of writing in a drawing in a paper, we have to use some tools here to get the networks. Okay. So coming to the next one. So now when you want to use the tool in order to create a network, we are dependent on some source or the data, right? So what you have to do is that you can go to the home page of NCBI. There you can type geo data sets. I'll be showing you, this is only the step for you how to go ahead. Then my area of interest, let me assume it is cancer. So in the search box, you can put cancer and you have to type over there as geo to R. Okay, so geo data set is to the R programming data. So that data set you can take it, and then under options you have to select certain parameters. One is bond Ferroni, as well as you have to set the log value. After that you have to even select your data for analysis, and after that you have to click reanalyze in the tool. Then the data will be whenever you're taking your cancer GO2 data set, 2R data set, check out how many samples are there. So the data set will give you the whole experimental design, what they have done, and is it an mRNA expression or what is the data. So you first understand what the data set is, and among them you try to take only the data set which is having more number of the samples in them. Okay, so that you will get a good network. That's the reason why. Then what you do is that after analyzing it, you will be getting a sheet which will be like an Excel sheet format. So you have to download that data set, save it in your file or your folder. The data what you have got will be saved in the form of TSV file. But what we require for further analyzing or creating a network must be in the CSV file, comma separated file it must be. So you have to convert your TSV into the CSV file. So it will come in your Excel sheet. So save that Excel sheet. After that, understand your data set. Understanding of your data is very, very important, participants, because you'll be getting thousands of genes also. So you should know which is the best one. You have to shortlist it, filter it, and take it for your further network process. And if you are very much interested, I want to take all and put an experiment, it means you can do it, but you have to wait for 24 hours or 48 hours to get you the output. So for today's purpose, we will take small parameters and work it out. And first you learn, then you can do the exercise by yourself. So whatever the Excel sheet you get, you have to rank them according to the log value. It is log FC value. So it's a fold change value. Based on that value, you will top and take the 10 set of the genes. For today's purpose, let us see, we are taking 10 set of genes. Now they have to be taken, and then you can upload it onto your string database. So the string database will generate one network. For whatever set of genes you have given, it will generate one network. With that network, you can transfer that network into your cytoscape, and further over there you can analyze your data. Okay, you can analyze the data even with string database, but some of the result or analysis, you won't be able to get a better output with this one. So we will go with the next advanced level of network analysis by using the Cytoscape. Okay. Now, this is how the network analysis generally goes on. You have to collect the genomic data, mostly it's protein-protein interaction, mRNA expression data related information from the data set has to be taken, and you have to sort the data. As I told you, thousands of informations will come. So your area of interest on what, which gene, what disease, what you want to work, you have to accordingly design and sort the data. Then you have to create the gene list. Then you have to understand the gene list, what you have selected, what pathway they are involved in. Is it a metabolic pathway? Is it a regulatory pathway? Is it involved in any other reactions inside the bio? So all understanding must be there initially when you are trying to use the data set. Okay. Then you have to visualize and identify the interacting pathways and the networks. Then finally, you can check the data to understand their molecular mechanism, because from large data set, you'll be screwing down and getting you the best output from your cytoscape, with which you can further try to understand what are those set of the important genes which are mainly interacting with the other proteins. Then you can either publish your data or you can create more models explaining the data can be generated. So the overall way of running this network analysis by using your cytoscape is take the network attributes, analyze, and if possible, if your data is reliable, if it is very good, you're very strong, your data can be published with the results or the outcome what you get from your cytoscape. That is the advantage of using this particular tool also. Now, as I told you that you are taking the data set from a particular source and then you are putting it into an string database, you will get the initial network. From that network, you're transferring it into the cytoscape and get the output. Okay. In that one, you can shortlist your hub genes. So that is the main significance about using your cytoscape. You can do multiple analysis, different analysis you can do, but I am going to talk only about the hub genes. Very simple procedure way of doing it. So what is this hub genes? They are the genes that have many interactions with the other genes are defined as the hub genes. If they have more interaction between the genes, it means they have high degree of important genes they are. If the interaction is less between them, it means the less important genes it might be. So what we can do, you will get lot amount of hub genes. So you can, when you are taking the source data and putting it, you can shortlist and take only 10 or 15 hub genes for your cytoscape tool. And with that one, you can initially start to understand what is the gene, what is the protein it is produced, and that protein is interacting with what other proteins. Are they doing upregulation or are they doing the down regulation? So all this mechanism involved by that particular protein in the network has to be understood by us thoroughly. Then only you can go to the next level. Okay. So in a protein-protein network, interaction network, when a protein has many edges, okay, connecting to other proteins. So now I told what is an edge. When I say protein, it means it is a node. It indicates that this protein interacts with large number of other proteins, signifying as a potential hub protein with a significant role in coordinating multiple cellular processes or pathways within the biological cell. Okay. So till this one I shared with you, I don't know how many of you have downloaded this into your system. If you don't have also, you don't have to worry about it. You just watch what I am doing. Till string database also you can do it. When time permits, you slowly can download the Cytoscape tool, because first thing is you have to download the Cytoscape tool. After that, you have to download some apps also. Then only you will be able to utilize the features that are present in the Cytoscape. Then only we can work to analyze the data set also. So is there anyone who have downloaded this tool in your system? Just one capital Y in the chat box. If no, means don't have to worry. If you are working with your normal system, you can do till your string database. Yeah, okay, okay. There are many of you. Very nice. Okay, okay, good. So others can work till string database. So you can, that also you'll get the network. How to improvise that one, I will tell you slowly. Okay, okay. Okay, many of them have downloaded it. Hope it was easy for you. So in your desktop, hope you are able to see the icon of this Cytoscape. Fine. Okay, fine. Okay, after this one, so what we are going to do is that we are going to go to the data set, take some information or the data set, and then you are going to use a string database initially in order to find the possible networks. Okay, now remember you're having a data set, you're taking some information from that one. So that becomes your source. Then you put it into your string database and then into your Cytoscape to find the multiple targets with which it is going to interact. Okay, so once you go to your Google, you can just, if you are working with your system, you can type the string database and you can access the site. So the search will get opened up. So if anybody is working, please carry on with this one. So under the search, you at present for this one, simple understanding with the pre-existing data set available in this tool itself, we will use and do. Then I will tell you how to get the data from a different data set and how to run it also, I will tell you. So just for understanding, let us do it. So, okay. So what you do is that under it, to your left hand side, you can see that there is protein by sequences, right? Or you can see a list of informations are here, or to my left hand side, protein by name, multiple proteins, protein by sequences are there. Maybe you have to select multiple proteins, and here what you do is under protein name, here examples are given. You see number 1, number 2, number 3. You take anything, you take any one. For example, if anybody is clicking number 1, there will be tryptophan operon genes list, or the proteins list will be coming out over here. Okay, and in this particular thing, if you select number 1, it will be coming as protein tript L or tript D, tript E. If I'm not wrong, this may come out. And by default, the organism directly will be selected as Escherichia coli, because operon we study with the E. coli. So here also you select only yes. Whatever by default it comes, you just take it up and you just click the button as search. Okay, so you can see the same thing I have done. So you put search, you will be getting the first output over here. Now try to understand the information what is given here. You took only 4 set of genes from the example given over there. So each and every gene, what it is being explained over here. So if you take trip A, it is the tryptophan synthase enzyme. If you take trip B, this is alpha subunit, this is beta subunit. So that is something interesting about this particular enzyme, tryptophan synthase, playing a role in the tryptophan operon. Then you have trip C, which is an indole 3-glycerol phosphate synthetase, as well as it produces 2 enzymes, and this particular pathway is required for the tryptophan biosynthesis in the E. coli organism. Then you have one more tript D, which is an anthranilate phosphoribosyl transferase gene. So that gene produces that particular protein. So how many you have taken? You have taken 4, right? So now what you do is that if you're working simultaneously, on to my right hand side here, you can see there is a word called as continue. Just click continue and wait. Some dots will come and the output will come within 1 or 2 seconds. Did everyone get this output for the 4 set of genes what you have taken? I think you must have got it. Thinking that you have got it. Now what you can do with this one? What is your understanding now? How many is the source data you have taken? How many you have taken is only 4. So how many nodes you have got on your interaction network picture? You got only 4, right? So you can see the names also reflected over there, and you can see a circle, and inside you're able to see some structure, right? That is the three-dimensional structure of that particular protein. If it is available inside, you will be able to see it. In case it is an unidentified protein, we don't know, but it is a protein, but three-dimensional structure is not being elucidated, then inside that ring, it will be empty. Okay. Now, you watch carefully the network. Did you find only one single edge between two nodes, or you're finding multiple edges between all these nodes? If I'm not wrong, everybody is able to observe that there are multiple edges between each and every gene, right? So now whenever you want to do the protein-protein interaction study or analyze the network and the interactions, it is always advisable to use more set of the genes rather than going with minimum set of the genes. Since you started with 4, you're getting a small network. But now let me assume that you want to increase the network. Very easy. What you do is that on to my right hand side, here you can find one plus symbol, right, written as more. Okay, just click once. Just click it once. If you click it once, wait for a few seconds, the network will increase. That means from the data available related to this particular genes or the proteins, some more genes are being added up as the node to this existing interactions. Okay, hope you got it. Okay, so like that you can go on increase. When I show it, I'll show you. So like this, what? But I started with the 4. Now you can see you can go on increase, increase, increase. By pressing the more button, you will get more number of interactions. So can you tell me what I asked you in the beginning? Take a piece of paper, draw the network and show me. Means if it is minimum, you can draw as much as possible. But once when the nodes are more, then it is very difficult for us to analyze. So we take the help of certain computational tools to do it. So you can see, so what I have done is that after that I have transported that network from the string database into the Cytoscape. This is the home page of Cytoscape. So there will be some number of nodes, edges, clustering coefficients, some data will be available on to your right hand side. Maybe when I'm showing it to you, I will show you all those things. Okay. So this is how the Cytoscape homepage looks like. So you have multiple parameters over here with which you have to check it out. You have file. I'm not able to take my laser point, so I'm just showing with my cursor. Here you have file, you have edit, you have view, select, layout, apps. So this is the one. If anybody is opening the Cytoscape tool, you can click the apps. There will be a lot of app store will be there. If you check that app store, list of apps will be coming out. Not all apps are free. So that also is very important. So only few are free online. So we have to install them and use it. Installation will take only 1 or 2 minutes. So whatever it is important, some apps I will tell you that time you can install it. Then you have the next one parameter called as the tools and the help, where you have a lot of tutorials with which you can understand in case if you are not able to perform any of your analysis. Okay. And next thing below, you can find the file where you can save your data. Then you can share the data, either import or export. And then you have a zoom in, zoom out, and the view symbols all are there. Then below that one, you have the network. Here I have enabled my string database so that the network Cytoscape can easily accept the data when I'm trying to transfer the data directly from the string database into the Cytoscape. In case if it is not active in yours, don't worry. We can download the CSV file. That CSV file you can import it over here and you can do your work. Okay. Then you have some other parameters over here with which you can play with it and try to change the node color, node size, then edge color, edge size or the shape of your node, anything you want to do, you on to my left hand side, you can see some of the parameters given like style, filter, annotation, and I have activated or enabled one of the tool which you are able to find the hub genes, which is called as a cytohubba. So this also has to be installed, then only we can do the network analysis. Okay, now in the middle you'll be having the workspace where already pre-existing some of the networks are available. So if you just click that one, it will come to your center page and you can expand, zoom in, zoom out, and try to understand what those nodes and edges are. And just below, if you go here, I'm able to find as node table. So if you have a network and if you go to your node table and if you expand it, you will be able to see all the individual set of nodes and their clustering coefficient or the organism from which it is coming. All the details relevant to that particular network will be listed out. So if you feel that this is one of your important information, you can download the data and save it also for your future purpose or working purpose. Okay, and so this is main thing. And what I have done, I have shown you some of the tools which I have tried to explore myself, and you can see I have got a tool over here like labeling all these things. You can border, you can change the font size. You can see from here you have to catch and you have to drag it out so that it will come out. I will show you how to do all those things. You can change the shape. I have enabled some of the features over here with which you can try to explore over here. Okay. So like this, I told now you can also, this is actually a layout, the layout of your network. When you're transferring the network from stringed to your cytoscape, in whatever format it was in string, it will come to your cytoscape. So once it has come, if you feel that that is not the way how it has to be represented, you want it in a circular way, then you have to go to your layout. You have the option over here, layout. There, if you click down, the scrolling down, it will come down and there you'll have a circular layout. They have tagged layout. So different layouts are there. So anything if you want, you can click and you can just change it out. That tries to help you to understand more better way of giving your data explanation to the others, or representation maybe much better, whichever you feel comfortable. I kept here as a circular layout. Okay. Now actually when I was transporting the data from the string to the cytoscape, the edges were all black in color, but I wanted to know whether I can change the color. You can see now I have changed the color to red also. And you can see that I have pulled out one of the genes from here, from the track. I pulled it out and you can see how many number of edges are there. And I am telling you now, it is your question and answer session. Now you all have to count and tell me how many number of edges are there. So can you do it, children or participants? I don't think so. Maybe if I have to zoom in, zoom out to check out, and the edges may be layering one with the other one because too many of them are there, right? So that is why in one of your tool, you'll be having a tool which is called as a network analyzer. So you have to click to that network analyzer. On to my right hand side, you can see that there is an output coming out of my network. Okay. Now you see what it is giving. How many number of nodes are there? There are 46 nodes are there. Now number of edges, how many are there? 170 is there. And average number of neighbors, what it is? Network diameter, network radius, then clustering coefficient, network density, and whatnot. So many parameters as an output will be coming out from your analyzer. Okay. So that is how, by using the cytoscape, you are able to initially understand how many nodes are there, how many edges are there. Okay. So it's a circular layout. Just now I told you. So this is the apps I told you. So some of the apps we have to take it out. So if you're working with your Cytoscape, if you opened it up, you can just click on apps, and then you can select, you can type over there a CytoHubba, and it will take you to the site directly, and from there you have to install it. Just a matter of 2 minutes, 1 or 2 minutes, it will get installed in your Cytoscape tool. Okay, string app is also there. If you want, you can enable it. Otherwise, not an issue. When I'm showing, you can watch it. Okay. So this is the one thing what I told you. From your app, from your tools, you'll be getting the analyze the network. So if you click this one, for the whatever network you have transported from your string to your Cytoscape, you will be getting your nodes, edges, and all the other details relevant to it. Okay. So now coming to the other things, now I told you there are so many number of nodes and the edges, right? So if you want to understand, you have to just drag the lower, you'll be having the panel. So hold that one with your mouse and drag it above. Down, you'll be getting all the details about the nodes when you select your node table. So I will show you when I'm working with that one. So you can see all the data. If you click one particular node onto your Cytoscape network, down you can find the relevant protein or the gene, and all the details will be given over there. Okay. So what I did was that I took one of the set of network, and I tried to create the hub genes for that one. So here in the CytoHubba is there. So after transporting your data from the string database to the Cytoscape, there you have to click your CytoHubba, and then just select 1 or 2 parameters. Ask for only, I've taken many number of data set over here. So you have to rank and ask only for top 10. So what it will do is that it will among the different set of important networks what you have taken for your study, it will top and rank and give you the first 10 important hit list will be coming out. And you can see that the Cytoscape has beautifully colored them, also ranking them from bright red color to the pale yellow color till the last one. So you can see on to my right hand side the string network with the ranking method. What I've taken is MCC. It is called as a multiple click centrality method, in which it has ranked the the genes what I have selected now. So top 10 has been coming out. So you can see in the network also some of the genes are only blue in color, but the ones which are being ranked, the best ones, are having a different color. And if you click that one, yeah, below the node table, you will come to know what is the gene or the protein. Okay. So then you can download, if you feel that this is one of your important work. For kind request to the participant, please mute yourself. So you have ranked, right? So out of so many genes, you have ranked them. So you can download it, and you can save your current rank also. Okay. Now you can see I have changed one of the style, or the one of the node shape also, right? All I've kept it in the oval shape, but since I find that according to my CytoHubba, this is telling that this is one of the hit gene or the hub genes in this particular ranking what I have got, or in this network what I have got. So that one I have changed the shape of it. So you can just double click on that one, go to your style. It will tell you the what to do. You can just change it. Okay. So that is how you can use this particular tool, and you can now analyze your networks. Okay. Is anyone has been working along with me when I was doing this, or happily watching what I was doing? Have you understood? Just put Y in the chat box so that I can take you to the tool, and I can show you how to do it. Are my participants there with me? Okay, fine. Okay. So shall I show you now? Still now, if you have any doubts, you can ask me so that we can go ahead with using the tool to check out. If you don't have Cytoscape, don't worry. You can run even with your string database. 50% of understanding will come there also. So don't have to worry about it. So shall we start? Maybe you're all working. So I'm going to hope my screen is visible. Stop sharing and then again I will share. Just a minute. Okay, so now I will share my screen. Okay, so I have kept the screen shared for you. Hope you're able to see it. Yes? Okay, so assuming that you're able to view my screen, I have taken here the string database, which is showing going to be used for the analysis. Hope you all have done this one till here. So here by default, I have, since I've been working with this one for some time, it's already preset with multiple proteins. So here you can select, as I told, you can go with your number one. I'm taking for you to be very simple. I'm taking number one, which is the source is what you have taken. This is your source. Okay. So now what you two can do is either you can give the list of names of all the proteins over here, or you can have a file also. So only having the list of genes, you can have them and you can upload it also. Okay. So as I told you earlier, tryptophan, we are going to, it has been studied only in the E. coli thoroughly. So that's why here by default, automatically it is preset as Escherichia coli. So we also will go that one initially to understand. So just go with search. Okay. So as I shown you earlier, same thing you are able to find trypt A, B, C and the D. Each one are the genes which are producing different enzymes which is required for the tryptophan operon. So what I said is you can go with the continue. So this is the output which I showed you also. So some understandings I will try to give you over here, which I couldn't do with because I did not save it over there. So as I told you, you can see that there are 4 genes and you can, so it is a 4 nodes and you can find the edges, right? Now, is the edge all are having the same color? Hope if I'm not wrong, all the edges are not having the same color, right? So what are all these things? You can try to understand by just clicking onto the legend. So you can just go down and see what each one is talking to you about. If your node is colored, so it's here red colored, it says that it's a query protein and the first shell of interactor. Then if you have a white node with the protein inside, it could be the second shell of interactors. And I told you, no, sometimes the node may be also empty. It indicates that it is a protein, but it is not having any, its structure has not yet been elucidated. On all the structures will be 3-dimensional structure. And if it is a filled node, it is a 3-dimensional structure of a known or a predicted protein. Okay. Now we just go below. I told you now there are so many edges between each and every node, right? So now what are they? If you go, you can see the word as edges, which represent protein-protein association. So one node is your protein, another one is your protein, how much of interactions they are having or association between both of them. So if you find a blue line between both of the nodes, then it is a curated database. If it is like this color, it is experimentally determined interaction between both the nodes. If it is this color, then it is I don't know whether you're able to see it. Okay, I've enlarged it. Now hope you are able to see it. So this is a green color. It's a gene neighborhood. This is the predicted interaction. Then this is the other color, gene fusion or gene co-occurrence or text mining. You have got this data or the genes from or co-expression or protein homology. So from the different databases available, the interaction between all the 4 set of proteins what you have taken has been shown to you in the form of a very small network. So if you just go below also, you'll be able to see the same data. What is the gene? What is the protein? It is able to produce the organism from where we have retrieved the sequence. So this is the legend. If you click from the string database, you'll be able to see it. If I expand are you able to see the three-dimensional structure of the protein present inside each and every node, right? Okay, now coming to the settings. So this is a setting which is like network type, what it is. We are not going to work much on this one because it's just the data already been preset. We'll just use the same thing. Okay. Now next one is another important data which you have to understand. That is called as the analysis. When I showed you your cytoscape, there also I showed you number of nodes, number of edges. So here also you will be getting number of nodes and number of edges. Okay. So now you can see this is the data, and as you go below, you'll be able to see the functional enrichments in your network also. So as you just go down below, more data related to that particular set of genes, what you have taken, you will be able to get the information. Okay. Now, next thing, what is called as the export. So if you click the word export, so you can see different informations coming out. You can see the data set over here. So this is what you want to require in order to further explore yourself or this particular set of genes you want to understand with the in a better network analysis by using cytoscape. So what you can do in case if you don't have in the cytoscape, if you don't have the string database tool enabled, don't have to worry. Just in here you have this thing called as download. See what is the download tool in which you have to download it. It is a tab separated values, can be opened in Excel as well as in the cytoscape. So this is the download format in which you are supposed to download the data set. Okay. So if you just click the download set, the data will go back, and in the downloads you can save the data. So it will be in the form of CSV file. So it will be in the form of TSV file, which is the file which will be recognized in your cytoscape. Okay. Then what you're going to do is that now, as I told you, in order to have more understanding about the network, just instead of working just with 4, we can amplify it. So here you have the more option. Okay. Uh, so we'll go to that one. One point you can understand, if you have enabled your string database in your cytoscape, then if you just click over here, send network to cytoscape, directly this network will go directly to your cytoscape. Okay. Then you have clusters, then you will be having the more option. So if you click more option, you can see that there is number of nodes getting increased. Initially it was 4 only, now it has become 9. So you can increase, add some more. So the node will increase to 14. So whatever data sets available in the database, so many proteins which can also interact with the 4 set of the source data what you have taken, it is trying to show you the possible network. So now the number has increased to 24. So maximum if you take, you will be getting the better hub genes. They are the potential genes which are having the maximum network with the other genes, okay, or other proteins. So you can increase it. So I have max. Yeah,
Ma'am,
Mahesh.
ma'am, actually we were given only 4 genes, right?
Yeah, that is your source.
Yes, ma'am. By
Yeah.
clicking more symbol, how it is increasing, ma'am?
Interacting. That means when you're taking only 4, the tripped in that E. coli, you have other proteins also present, right? So it is trying to see whether this particular tripped is able to, this particular protein is able to interact with various other genes which you have not taken. You have not taken, but it may interact with some other proteins, right? So when you are putting it more and more, the program will be designed in such a way that this particular gene, in case if that particular protein is having any other gene or any other protein interaction, automatically it will take that say data and it will show you whether the interaction is there or not. So it's automatically taking up and increasing the number of interactions of those 4 set of genes with other multiple genes also present in that particular organism which you have taken for the study. Okay. Yes.
Okay, ma'am.
So now I have amplified the data. So what is the number of nodes over here? 44. So number of edges also have magnified up to 434 number of edges. Okay. So now what you have to do is that you have to enable which one now? The cytoscape, so that you can transfer this data. So you can click onto the cytoscape. So the version, I hope everyone has taken 3.10.3. That is the latest version which is available in the cytoscape site. So if you have opened it up and if you have transported it, well and fine. Have you downloaded the data? If you have downloaded, then you have to transfer the data. Okay. So you have taken the source and you have got the targets. Okay. So now you are going to use the, so go back into your string database and I'm going to click on export. So I'm sharing the screen. Hope it is visible. I'm asking now my network to be sent to the cytoscape. Since I have already downloaded it and my string database is enabled over there, hopefully this network will get transported to the cytoscape. Okay. So it is taking just some time. So
Ma'am, in the exports, we have two CSV files, right, ma'am?
1 minute. I will open my cytoscape, then I'll come to that one. It is sending the network now. Once it is sent, I'll come to cytoscape. That time I will tell you. So network is sent. It is showing me here. So I will open this network. You can see the network in my tool. Okay. Now what is it, Mahesh?
In exports,
Mahesh?
ma'am, we have two CSV files, ma'am.
First of all, you have downloaded your data, right?
Yes, ma'am.
Everything you cannot send it to the cytoscape. You have the genes, right? You have gene ID symbol, something will be there in your cytoscape downloaded tool, right?
Yes, ma'am.
You copy some.
Okay.
And then paste it over here. In the network, you can paste it. I mean, here you can paste it. If you paste it, you will be able to see. You have to select directly. You cannot transfer the file. You have to select some genes and then you have to transport it here. If you have a log value, you can sort it out. Selecting the one which is having the highest log value, I can transfer it. Or at present, select some genes and then you transport it here. Are you able to do it? Okay, so now I am able to transport here. You can see that one of the, protein is empty also, the shell, that is a node. So now what you can do with your mouse, you can hold it and you can remove, because I find many number of overlappings of the nodes, right? So you can separate so that you can see if anything is hidden in between, you will be able to view them. Just with your mouse, you can just try to separate them. Are you able to do it? Any strucking means you please ask me. So here overlaying, I'm trying to separate the nodes. Okay. So here is one overlaying. Okay. So maximum, I think if I'm not wrong, I have tried to show you the nodes. Okay. You can see how much of network is there. Now first thing is, let us try to do the network analyzer, which I told you. So if you go to the tools, you can see your network analyzer, click that one. Okay. So it is asking whether you want a directed graph or whether you want it a undirected graph also will be there. By default, I am selecting only as directed graph and giving it as OK. Okay. So now here you can see the data, summary statistics. How many number of nodes have been taken? String database, if you check, it will be 44. Here also it is only 44. The edges are 434. And average number of neighbors, network diameter, and other parameters what is being available, you can have a look at it. Okay. Then coming to the next one, below you have to go down. Okay. So you have this below panel, right? So this is your showing the grid down below, then show the view. All those things are there. So let me show, just pull it up. Then you can see here, if you select any one protein, that particular protein ka data will be coming out. So here you can find the node table. Just click that one. If you say node table, it will give you the node table. Okay. Then if you click edge table, it will give you the edges, all the possible edges between all the interactions, the data sets will be going on. So if you want to export it, you have the enabling tool also to export the table to a file format, and you can save it and keep for further analyzing the data. Okay. Then network table, if you take also, you'll be getting the string network table. So you have to just work this one and check out all your data sets, what is there about the each and every output what you have got. Okay. So this is one thing what you can do. So you have transported successfully the network from your string into your cytoscape, and you have got some set of nodes and the edges. So what we can do is that with this available data, let us see how to get your hub genes. So here, as I've enabled the tool, I'm just clicking onto the Cyto Hubba. If you go to your apps, if you go to apps, App Store, show App Store, you can see all the list of um, apps will be available. So some are free, some are paid things. So you have to enable them. So you have to download it from their site. Okay. So this also, this Cyto does not come with your Cytoscape. So because nearly about 300 different apps are being created by the Cytoscape, in different versions. So according to the requirement for the analysis, we have to go for the downloading of it. Some are free, some are not free. So depending on your requirement, we have to enable these tools in your Cytoscape. Okay. So since I have enabled this particular tool for the study, I am going to click here. Now here already we have taken the string network only, which we have imported. So automatically it has taken up the target network. Okay. Then you have to press the node scores to calculate and export it. Now here is the Hubba nodes where you have to specify how many number of nodes you want. You can take 10. You can take 20 also, but generally preferable is to take the top 10 or the 15, where you can see the good ranking or interactions initially between them. So you can start from 5, then you can go to 10, then you go to 15, get the output, download all the data sets what you get, and then you try to analyze with what is the sort of interaction, what pathway this protein and that protein is involved. Like that, individually we are supposed to analyze the data. Okay. The next thing is MCC is given here. So when you scroll it down, there are various methods in which you can get your output. For convenience, let me put here as degree. So then you can go here and display options. You can check the first stage nodes, display the shortest path, and the expanded subnetwork. I'm just putting it as submit. Let me see what has happened now. So something has happened. So what has happened here, you're able to see now, right? So there is so much of output coming here. So now I'll go to the analyzer and convert from string into the Cyto result. Okay. So now you see it has been ranked, but what has happened is that too much of network for your up genes. So just try to separate them from the other genes which are there. So just use, since we have selected 10, expect for the 10 set of hub genes to come out from your cytoscape, and then you can check the ranks. Okay. I maximum try to get the output. Okay. Now coming to your first one, here it is ranked 1, right? So there are so many red over here, but I find 2 red which are bright in color. So I want to know which is the among these 2 which is ranked 1. So you click on that one, and down panel if you go, you should be able to see the information about that particular one. So you can see it is from the string, it is from Escherichia coli, and you are getting it from the string database. Okay. So network table is enabled, convert it to node table, then you'll get the data. So you see it is a shared name, canonical name, then you find the node color. They have different colors are given, name numberings are given. Display name, what gene it is? TRPD gene. That is a target gene, what you have, that is source gene, what you have taken initially. So that is seeming to be having the one of the hub genes, one of the main gene or the protein which is now able to interact with multiple other proteins, forming a huge network. Okay, so then you can go to the what is the expansion, what is that a particular protein it is able to produce. Just go downwards. If you just expand, you'll get anthranilate phosphoribosyl transferase. You can see all the details coming out when I am going to select the protein tag. Okay, so if you feel this is important for me, you can export the data. Okay. Then another thing, since here they are given the numbers which are the accession numbers from where your protein has been retrieved, you can rename this. Now only we saw what this gene is, right? So do the right click on this, go to edit and rename it. What is the name you have found it as? TRPD. So just rename it and give it as OK. Automatically the name will be renamed. Okay. Now I'm not still happy with it, because once I find that this is my hub gene, I should make this to be visible remarkably when compared to the others. Most of the time when they are trying to get the hub genes, the ranking are being given the color range, right, from top rank till the least interacting proteins. So instead of changing the color, it's better that you change the style of it. So you select, double click on to your TRP1 and go to the style. So already what it is telling to you is that this is all the parameters with which you can try to change that one. So already here it is filled with what color? It is filled with red color. Okay, so what you can do is you can change the shape of it. So if you click that one, multiple shapes are available. Maybe I'm interested in diamond, so just put apply, and you can see all have got become this one. Okay, click this one, and for that one let us change the shape. Eclipse. Okay, all are changing. So maybe if okay, so like that you can do. If you want to change your edges also, you will be able to do it. Okay, so this is one parameter what you can understand with it. You can change the shape also. Now what I'll do is, this is one way of representing your network, but still what has happened is the other genes what has been taken are mingled with the other one. If you want to use a data set, this representation for any of your study purpose, then make sure that all the nodes are visible. So make sure that you separate them out because they are laid one above the other. So you can separate them out. Okay. Then what I can do is that I can change even the layouts I told you. So I will show you. So now maximum I try to show in a better way all the different nodes. Hope if you count according to the network analyzer, 44 must be the nodes. Okay. Now also, so go to the layout, and you can see you have the grid layout. So all nodes you can gets into the grid format. So you can see how they are arranged. Okay, then again into the layout, if you go to hierarchical layout, if you select the nodes and do it, that's also okay. like selecting. So this is another way of representation, very difficult to understand, but maybe let us see what happens. See this now. Okay, so now this is a circular layout. You can see how the edges are there. Edges also tells you the distance or the interaction. I told you earlier the example. Close will be there, some will be distant, some will be a little bit far away, right? So depending on one node connecting with the another node will not be of the same distance. The distance will be different. So that also will be shown in the form of those enabled tool here. You can see them. So if you want, you can separate your hub gene separately from your circular pattern. If you feel, no ma'am, this is fine for me, it is looking pretty enough, fine, it's up to you. Okay. Then stacked node layout. This is stacking. One below the other is being stacked, and in between you can find the hub genes, and they are being stacked. So you can just remove them and you can do like this. So that's what, once you are having the tool, if you know basically how to use the tool, then you can just do multiple things with this particular tool. Okay. So any network, if you want to, you can start to do. Okay. So what we have done till now is we have tried to take an example of the pre-existing data and then use it in the string database available and put it into the cytoscape to get a network and find the hub genes. Okay. But let us assume you want to work something, a new one. You want to work, think something new, innovative, something interesting thing you want to do means, then I will tell you how to do that one also. So hope you are able to do this one. Yes, anybody has tried it out? Anyone has tried? Just give me an S in the chat box.
Ma'am,
Yes.
ma'am, it's with Prakriti.
Pardon?
I am unable to access the chat box.
Okay, parvalలేదు. You tell me what it is, Prakriti. Is it a problem or is it okay for you?
No, ma'am, I followed what you did right now. I'm
Okay.
unable to sit in the row that went, ma'am. The row one, last one that you showed me. How to put the nodes in.
Stacking. Go
Yes,
to the
ma'am. Yes, ma'am.
layout. Go to the layout. Go to the layout. Here in the layout you have, right, stacked node. Select
Aye.
all nodes. It will get stacked.
Yes,
You didn't
ma'am.
get it?
Yes,
Got
ma'am.
it?
Got it. Got it.
Fine,
Yes,
fine.
ma'am.
Okay, okay.
Thank you, ma'am.
Yeah. So you can select the nodes. You can select all the nodes, and I told na you can color them, change them. You can do. You can just try to work with this tool. Multiple features are there in this tool. That is not possible with your string database. You may get the network. But what I did now, changing the layout, changing the colors, changing the shape, if anything you want to do, you should be able to do with this tool. Okay. So that is the added advantage with your cytoscape. Okay. So I'll just wait for a minute. If anybody is having any query or not able to, you can just ask me. Otherwise, I'll proceed with the next step in case if you, this is with the known data, we have been running it, right? Something new you want to try, then what to do with that one. That also I will tell you so that you can work it out simultaneously. Shall we proceed? Means you should put capital P in the chat box. I am waiting. Proceed means next. Okay. Okay. Or you want some 1 or 2 minutes, something to play with your cytoscape. If you are doing 1 minute, 2 minutes, I will give you, then we can proceed. Okay. So 3 people are saying proceed out of so many people who are there as my participants, right? I need active participation. If you're having a system, no cytoscape, I said you can do till string database, right? You just increase the nodes, try to understand, can we specify a particular protein-protein interaction, a specific condition like a specific, yes, that's what I will show you now. Maybe not a particular, if you want to get a particular protein-protein interaction, specific disease condition, I will tell you how to take it and do it. I will tell you now. Okay, so I am taking it as a positive note to proceed further. So I'll just go. You work along with me. I'll just go step by step. This is right. Stop sharing. Okay, and then I'll take a new tab. In the new tab, I have not yet shared. I'm just clicking new tab. I will just show it to you because my Microsoft Teams is not okay. I'm sharing it now. Hope your new Google site have opened up. Just to say why. Just to say why. I hope you are able to view my screen. Yes, okay, fine. I'm assuming that you're able to view me. That means the thing. Okay, now in the Google, you type it as geo data set. Okay, have you typed it? So just click that one. It is an NCBI geo data set. I'm giving you one set of because there are a lot of data which will be available. So to make it our work easy, I'm just giving you one particular geo data set. You also can type the same thing to retrieve the data. It is GSE 22439. I'm holding it for a second. Under the geo data set, you have to search for a set which is GSE 224309. Okay, now you can go for search. Okay, so I am getting an output over here. Just check out your accession number over here, whether what I've typed, it is the same. It is the same. Okay, now we're going to work with this one. Now, what is the number of samples you see? 16 are there. That is why I wanted to select this one. Second thing is that when you are giving this GSE and giving the number, when you get the data set out, you please check another parameter what is available for this. That is analyze with Geo2R. So this has to be available with your data set, whatever you're retrieving from the geo data set. So that is why when you're typing also, you can put cancer and then you can put as GEO2R. Somebody asked me, right, for a disease state. I have selected a diseased condition only for cancer. You can see the title of that topic, right? So it is a bladder cancer, which is one of the most common cancer type worldwide and is characterized by high rate of recurrence and all. So the organism in which they've studied is the Homo sapiens, and the type of data what we are going to do the work is the expression profiling data obtained by the array technique. Okay, so 16 different sample data set is available over here. So we can take this one. Got it? So hope you are there with me. Now what you do is that now you click on this analyze with Geo2R. Geo2R means it is moving from the geo data set into your R programming. Okay, so now you have got the output over here. So you can just have a look at the data set here. You have the source name, you have the cell lines on which it's in vitro cell line study. And this is a cell type on which they have studied, transfection plasmid. All the data is available. And if you just go through, I told it's only 16 data sets are there. So now if you go to the data set, the thing is that if you see this one, this is check the data set, 16 are there, right? Some of them are urothelial cell. The study is done on the benign urothelial cell. Okay, so till here the data set is for urothelial cell, and down if you go, it is for the bladder cancer. Okay, so that is why that's why I wanted you also to take this one and work with this one if possible. Okay, so now just to understand you, now this is not the thing what you're going to exactly do. Now here you have defined the groups. Okay, so please click that one and you have to give the name for the group. So I told two groups are there, 16 samples, two set of group of data is there. So you go with one urothelial cell. Okay, and press enter. So you can see that down it will come. Click this one and you're going to take the sample set. Now, when I click that one, it is showing 8 samples, but it is showing me down the selected one is cancer, bladder cancer. So you go to your urothelial, put your finger on your control and select only the urothelial cell. So you have to select the data. Okay. And then click, it will take urothelial cell, but it has taken 16. Okay. So do you have to repeat it again? Because earlier we typed it. Okay. You can do this one. So go to this and with your control button, you just select all the urothelial cells on which the study has been done. Then you go to the urothelial cell and click it. It will show now with that I have selected the from the data set out of 16 samples, 8 samples, 11 to urothelial cell has been selected. Okay. Now again you go to your define the group and put as bladder cancer cell. Okay. And then click enter. Okay, so for that one, what you will do is that you deselect or no, okay, go to this one and you put your finger onto your mouse and or your control and select all the cancer cells. Okay, I selected here. It is showing 8. So now what has happened here is that you have selected or you have separated your data or filtered your data into 2 different data sets, urothelial cell and the bladder cancer cell. Here you have 8 samples, here you have the 8 samples. Okay. After that, you go to an option file over here and click it. So this is the different parameter settings are there. One or two we will just set, other one is auto setting only. So first one you select as Bonferroni, which is a tool to determine the p-values. And you can set your log value here over here as 1. You can make it as 2 also, or you can make it as 0.5 or whatever you want to, but not more than 2. And then you can go with reanalyze the data. So it goes for processing. So the data will be analyzed. So whatever data sets you have selected for urothelial and the cancer, whatever genes or the proteins with the log value, the output will be generated out. So this is the output file, what it has given you. It's a big file which has come out. So you can see that you have the ID, you have the p-value, the adjusted t-value, then the b, then the log fc values, the gene symbol is there, and then you have the gene title. So we are more concerned only with the gene symbol. So if you want to directly use your cytoscape also, take some set of your genes and you can work with that one. But this particular file is not in the required format. Anyway, we will say what to do. So download this full table. So it's downloading it and it's given for me as table number 3. I'm saving it in my downloads only. Okay. So now this file is what file? It is your TSV file. So when you are using your cytoscape, you have to convert the CSV into the TSV into the CSV file. So what we can do is that you can use your online tools to convert TSV to the CSV file. So take this one and browse the file. Just now, whatever I downloaded the TSV, just open the file for it. Let it get uploaded. Processing will start. And then you can ask it to convert it now. So convert the TSV into the CSV because you have to you have a many set of data, right? You can't give all the data into your cytoscape or into your string. So we have to shortlist it. So for that reason, you have to select only the important genes in this particular process. Okay. So now you can see the file is Microsoft Excel, comma separated values file. That is a CSV file. I'm saving it in the downloads only. Okay. So now I'm opening my downloads.
Ma'am, Can you please repeat how to select the data to convert it
How
into
to
a
select the
pixel?
data? Okay,
Yeah.
fine. Okay. Minimize this. Go to your geo data set. Did you go? Where are you stuck? You tell
Yes,
me
ma'am.
which.
I have like I am looking at the table, ma'am, with the
This
urothelial
table
cells.
you got?
Previous one.
This one or the previous one?
Previous one, ma'am.
This one?
Yes, ma'am. Yes, ma'am.
So what you do is that here you have defined groups, right?
Yes, ma'am.
Click that one. Small
Yes,
window
ma'am.
will open up. Here it
Yes,
is
ma'am.
asking enter a group name. You want to group your data into one set, right? So only
Yes, ma'am.
two data is there. So you type the name. One you type it as urothelial cell
Urothelial. Yes, ma'am.
and go to your sheet and select urothelial cells alone. How much ever you're selecting, that number
Yes,
should
ma'am.
be reflected here. If you're
Yes,
selected
ma'am.
8, you should show
Yes.
8 here. Finished now. Now you go back again to your enter a group name. You type over that bladder cancer. And then go down, select only the bladder cancer cell type. And you should be able to see the cell selected type to be as 8. 16 samples, 8 of this one, 8 of that one you will select.
Yes, ma'am.
Able to. Now you go down and go to your options. There
Yes,
you
ma'am.
keep one of the option as Bonferroni, and also you can use your log as 2. Fix it as 2. If you want with 1, okay. Sorry, one I'm fixing here. 1 you can type and just go for reanalyze. It will sort the data and take all the informations what you have asked for, the log value, all those things will come. Is it okay?
I did not understand. Oh, Bonferroni. Okay.
I'll come to that one later. We'll come to that one later. That's a tool, right?
Yes,
We'll come
ma'am.
to that one later.
Log
Okay.
value should be 2.
2, you fix it as 1 currently now.
Yes, ma'am.
Okay, don't go for the maximum. Okay, so my Excel sheet has come out over here. So you can see whatever data you can retrieve from that one, you will be able to get your values. So my more important concerned are 2 different informations from this one. You have to filter your data and take only 2 sets. One, you take log FC value. Log FC value is a log fold change value. It is a logarithmic value. Okay. And it shows you the difference in the gene expression between the 2 groups what you have taken. That is the significance about your log FC value. So that is what I also wanted. So that is why you select the log FC data. Okay. And then you select the symbol against that one. Okay. Copy those 2 alone and go to the next sheet and you just paste it. Okay, so you have selected only 2 set of informations over here. One is your log FC value and your gene symbol. Okay, now what you do? You have log FC value. If I just go down, you have so much of data related to the log FC value. Some are negative also, right? But out of this, I cannot, it's a huge data set. So I cannot take all the genes and put it into my string or into my CytoHubba. Okay. So what we can do for our understanding currently is you have to filter your FC value. Go for the FC value which is highest, indicating that it is having the more interaction for a expression, gene expression. Okay. So that one you can sort it from largest to smallest. The data gets sorted out. Okay, so now you can see log FC value highest going down below is the smallest values you're getting. Okay, now what you do at present, forget about your, we have used the FC value to get your value of the gene which is having the highest log FC value. Okay, so now you come to your gene symbol, select your genes, maybe 10 or 15 or 20 you can select. maybe I will go with some 30 set of genes so that we can create network with this genes. Okay. Maybe you can open one more Excel sheet and just paste it so that I will do the analysis. Okay. So I have taken these genes. Okay. Some genes I have taken. So now you understood from where you can get or retrieve your genes. I've taken cancer. You want to take something else also you can take. Okay. Copy the genes. Okay. And then you can go back again to your string database. Okay, here is my string database. So I can go back. Okay. And freshly I'll start. Now you can paste your genes here. Okay. Now you have to select. This experiment was not done with E. coli, right? It was done with your Homo sapiens. So you have to give us Homo sapiens. Okay. After that, selecting Homo sapiens. Okay, so we have given the list of selected genes only, and then you have selected your organism. Now you go for your search because we have taken some set of genes. So now you can try to understand what these genes are. One is FOLR1, folate receptor alpha. Then it is folate receptor alpha, which binds to what they do. So they are showing you the list of the proteins. Then you can go with continue. The network will be coming out. So there is a small network which has been generated. So what we will do is that if you want, you can increase, but since we have selected only this, we will go with this one only into your CytoHubba and also see, is there any possibility to have the hub genes among these what you have taken. Okay. So I'm trying to send it to the network to Cytoscape. So that is why, so the network is very poor or weak. So that's why you should take more set of the genes for interaction. Okay. So for convenience sake to show, I just took only some. Maybe if you want to, you can do it more. So this data has been come to my set. You can see here. So this is what has come. So let me try to go to CytoHubba, and instead of string network only has been selected. Export instead of 10 because it's very much minimum. Let me go with 3 top genes, possibly whether it is able to give. Let me submit and see. Okay, a minute. What I have done is from the net says you have to select your network. The network what was previously existing by default it got selected. So I changed the string network to 1. Now you are able to see the network. Calculate and export. So here you can put as, because it's very less, I can put as 3 and submit and check whether I'm able to get. So I got it. So here also I kept the degree only. So I've got the set of networks. So among all the set of interacting proteins, only one is having the higher order ranking. Because why I said higher order of ranking is reason is because this only is red in color, other two are yellow in color. So interactions are there, the proteins are interacting, but they are not very close, or interaction is not very strong when compared to that, or the network is very less when compared to this one. Okay, so now what you can do is that you can save this current rank also, click that one. So again you can go to your downloads and you can put it as ranking 1 and you can save the document. Okay, so when you go to your downloads you'll be able to retrieve the Excel sheet, all the genes and the proteins and what is the number of networks it will come. I'll show you after some time. So now what we can do is that you can select this one and go down and check out what is this particular gene on protein, what it is going to produce. So I selected, what you select here also is important. Node table you have to select, because I've selected this particular node and you can see what it is. So if I go, it's a protein, it is by definition. Now if I expand, you can see 2' 5' oligoadenylate synthase like protein. So some different sort of protein, and all the other details related to that protein is available in this node. Okay. And the display name is QASL. So I want to demarcate or differentiate this particular node from the other node. So you can also put it as, I hope it is OOASL, and you can say it as OK. So it has been renamed. Okay, so like this you can get, if you click and want to go for edge table also is giving you all the protein interactions. Just click on any one, and if you just try to see all the scores from where you got the data, also is given as text mining. So you can try to explore with this one. Okay, so that is one thing. Okay, then if you want to go with your analyzer also, let us check for the analyzer. Maybe I'll go to the apps and then check. I'm sorry, tools, analyzer network, directed. Some are directed, some are not directed. So it is saying you the number of nodes are 7, number of edges are 10. Okay. So among this, we have ranked and we got that out of the 7 nodes, one of the node is the best node having good number of interaction with the other nodes for what you have taken for your study purpose, or for this running purpose, what we have taken. Okay. So this is what I thought I will share with you. So now if you have any queries or if you got stuck anywhere, you can please ask me. Anybody has worked till here? Were you able to go to the geo data set? You got stuck somewhere, you can ask me for it. How to get the accession number of our 4 genes of interest for a specific disease for geo data set. Down you have your ID number. When you're taking your Excel sheet, download is there. ID number is there, you see. Click that ID number. It will take you to the site from where the data has come. From there, you will be able to get the accession number. Yes, anybody has worked? Can you, if you have worked, you got some output string database or with your Cytoscape, I'll be happy to see the screen of yours if you have worked along with me. No one has worked along with me for today. I'll stop sharing so that I'll make it comfortable for you. How is the edge table below can be seen in the expanded form, ma'am? Okay, so now this is one, right? Is this looking like this, right? Now you have your below one ribbon is there right here. Okay, that you with your mouse, you can see now it has come like arrow mark on both directions, right? Forward, backward. Hold that one and pull it above. Okay, then you find here you can find edge table, right? So here you click node table.
You have to share
Whatever.
screen, Vanita.
I'm sorry. I forgot. I did stop sharing. I forgot. Sorry. Okay, now I'm sharing it. Hope it is visible. Okay, I'll go back again what I did. Okay, so this is how your site might be looking to you. So what you do is that here you have this panel, right? This ribbon is there. Hold that ribbon with your mouse. You drag it above. Then here you can see the feature which is called node table. Click that one. If it want node table, click node. So you have taken 7 nodes, right? 1, 2, 3, 4, 5, 6, 7 datas are available. So you will get all the data for that. You can find here also some numbers. You can take those numbers, ESPN, ensemble, protein numbers are there. You can use this one also. Then you have the other datas also, string database, all those things are there. If you click this one, you can get edge table. Hope whoever asked me, you're able to do it now. Yes? It. Any other query in my chat box? Got it. Okay. No one is going to share the screen with me. Anyone, or you take a screenshot of it and put the image of your network into the chat box. If that is possible, okay, fine. Should I wait? We should appreciate the participant because they don't disturb the resource person. You have to disturb, right? Okay, if you have understood, if you are clear in what I said, just type U, capital U. Yeah, okay. I'm getting Hubgenes also. Oh, that is nice. Whose it is? Oh, very nice. You can rename the top hit gene Samma, whoever it is. Nice, that's good. Okay. This is okay. Good. Okay, fine. Network is less, but maybe you can increase your network also. So you can, now I showed you how to go ahead, right? So you can try with different set of data. So I just typed cancer. I got this one data set, which was good. So I am showing it to you. So like that, you can specify the cancer. If no, ma'am, I don't want all these things. I want a better reliable source means you look into your articles or journals where you will be able to take what is the disease, what are the genes which are upregulated, what are the genes which are downregulated. So that one you can go to the literature and get the reliable data, because when we do the network analysis, you can do as directed analysis, undirected analysis. Directed analysis is like between both the proteins, you're going to check how much moving, whether it is from this one to that one or from that one to this one. Undirected means between any two, you're not having any understanding about it, but there are two networks between them. So you have directed in undirected. So many other illustrations and examples are there. So somebody is asking me, please illustrate how to change the node colors. Yes, I am sharing the screen now. Okay. I will go back to my network. So if this is my network, so you just select one, select this one, okay, and you go to style, okay, and there you can see color. So you can yellow, I'll put it with green and see whether I'm getting it. Oh, it's not changing. Label color. Okay. Okay, here fill color. Sorry. Here you can go with fill color option. Maybe this, if you can enable it. Let me see whether I'm getting with this one. If I say okay, no, it is not changing. It's not changing. Might be you can just try it, or maybe this is the fill color option. Just I will check it out. This is a shape. Now it has changed, right? The color has changed. This one yellow has changed. Maybe it has taken some time, but you can see now the color has changed. Maybe you have to go with fill color. You just try it out. Whoever has asked me, you can just try it out. So it is showing filled, but let us see whether it is filling. It has changed to green. Okay. You just have to check it out. So it will work out. Colors will come out. I have tried with that one, but now maybe I'm not able to, but maybe you just check it out, you'll be able to get it. Just select the node, double click on the node and go and fill color. You select that option. It should change. It will change. Okay. Any other troubleshooting? So if you want any other data set, you can go and search and you can get the reliable data set other than the geo data set, what I said. So you can take it from the sources like journals or articles who are working with cancer. So if they're working with the cancer, like mRNA involved expression analysis, they have done microarray analysis, they have done means from there you will be obviously getting some set of genes, right? So you can take all those genes from their supplementary sources, then you can use for your, that is your main source. Then you take that one and try to see whether there are interactions between your own genes or is it having interactions with the other genes also. So that is how you increase your node. So when you increase your nodes, not only this one, this particular genes, whether they're having interaction with other genes also, you'll be able to explore. But step by step you have to go, because if you, it looks very pretty to go on increase the network, but analysis part you have to be very careful. Now to show you, I've shown you only little of genes what we have taken and we have worked out with, but we have left so many genes behind, right? We don't know that those genes also might be amongst them there may be some hub genes, even though the p-value was we have ranked them according to p-value and we have done it. We don't know that some other genes present in your output also may be able to interact with your other proteins, right? So it's like trial and error multiple times with different set of genes. You have to work it out so that whatever output you get, you have to analyze it and give a solid information, a data which will be an authentical proof telling that yes, this is the protein taken from so and so organism having an interaction with so many other proteins. So again, you have to go back to literature and you have to check what you have got as an output from this cytoscape. Is there any data relevant to that statement? What you are saying is available in the other sources also. So if you are getting it, then you may have a very good understanding of the analysis or the network what you have got. So actually we have used the cytoscape by using with eugenol, because you can work even with certain compounds, phytochemicals also. So we have taken eugenol, and eugenol is present in the osmum sanctum, that is your tulsi plant. So we have taken that structure. You have different formats in which you give. So this is not a protein. So we wanted to understand this eugenol, what are the proteins it is able to interact with, or it is inducing what protein-protein interaction. So we have taken the SMILES notation of this particular eugenol, and then we have created a string database with that one. And that was imported to your cytoscape, and we have tried to analyze what are the proteins which it is able to create a network. Okay, so like that, it's like step-by-step process. Once you know how to use tools, mostly you must be able to try to explore it further later. Okay. So thank you all for your patient listening. Hope the outcome of this course is to make you to learn something. Maybe not 100%, at least 50 to 60% of today might be you must have explored yourself or try to understand what is protein-protein interaction and how to get a network and how to basically do a small analysis with the network what we have got. Okay, so thank you all. Over to you, Manju ma'am.
Thank you, Dr. Vanitha ma'am. Thank you for beautifully illustrating the protein networks using the GO2R string as well as the Cytoscape tools. So I hope all the participants understood the way how the proteins are interacting. So you can try out the various these tools and thereby explore these tools for the protein networks. So now I request to please post the attendance form, ma'am. So, and if anybody is still having any queries, please either unmute yourself or put it in the chat box.
The attendance sheet is posted. If any query is there, you can stay back. If you are finished, done for today, good night and goodbye for the day.
Once again, I extend my sincere thanks to the resource person for today, Dr. S. Vanitha ma'am, and I thank all the participants also for actively participating in this session. Thank you, ma'am.
Thank you.
Vanitha ma'am,
Maybe
thank
till
you.
you- Yes, Kamala ma'am, thank you so much.
So I
Prakriti,
request
are you
all
there?
Sorry, ma'am.
Madam,
Prakriti was asking me doubt, so I thought I'll speak to her. I'll stop the recording.
Okay. Ma'am.
I tried to follow as much as possible, ma'am.